Good morning. What I was asked to talk about today in the context of the validation discussion is a little bit about the role of validation of process controls, particularly those process controls that are critical control points, maybe OPRPs in FSSC 22000 schemes, or other types of control measures that are in our processes very important for the management of hazards in our foods. I did want to do a little bit of theory. Nancy gave a good discussion of how to set up a validation study, so I want to review a little bit of that, talk about some of the resources and challenges, talk also about the approaches that we need to take looking at the control measures, the various control measures in our systems, and then give a couple of examples of some of the things that we're talking about in practice. So the first thing I want to start with, because I always want to start with the definition of validation and verification. Anybody who's taught HACCP before, or taught food safety management topics before knows that this is often a point of confusion, and if you throw in monitoring, it can cause even more confusion. So what we're talking about in validation, and this is the Codex definition, is obtaining evidence that a control measure or combination of control measures, I want to emphasize that, if properly implemented, is capable of controlling the hazard to a specified outcome. So this is looking forward. This is saying, can my system work as intended to control the hazards I've identified? Verification is the application of methods, procedures, tests, and other evaluations in addition to monitoring to determine whether a control measure is or has been operating as intended. So this is backward looking. This is saying, has my process been working? And of course, also included as monitoring are those things that you do during the process to make sure your process is under control. So in this talk, we're talking specifically about validation, those things that we do that are forward-thinking, talking about how my control measures or my combination of control measures work. So in food safety system, we know that validation is not just limited to CCPs. We know we have other types of control measures in our food safety programs that are also responsible either to support a CCP or to work in combination to manage the hazards that we've identified in our food. And it's important that we know all of those control measures, either individually, if that's how they're in our system, or in combination are working as intended. We need to also validate that we've selected the right parameters, we have the right process controls identified, that we're managing our system appropriately, or that control measure appropriately. We need to validate how we monitor that to make sure that's the appropriate way to monitor that. And also we need to validate our verification. We need to know that the way that we verify that system is sufficient to ensure that that system is under control. So the scope and applicability of validation really depends on the nature of the control and the availability of tools, as some of the previous speakers have mentioned. And some of the effectiveness of some controls, in fact, are more effectively evaluated as an evaluation through ongoing verification because they're not really so suitable for the kinds of validation that we look as a forward-thinking evaluation of our process. And I want to give you a couple examples of that. And these are from some of the discussions we've had on the Food Safety Modernization Act based upon our experience with some of the food safety systems we have in our operations. And you see there's certain types of controls that do manage the incoming risk of raw materials or the risk of recontamination. And some of these, like hand washing, you don't really validate hand washing. You might validate the sanitizer that you're using or ask that your vendor has validated the sanitizer, but really you verify in an ongoing basis whether your procedures work. So similarly, there's many points in our process, there's many types of controls in our system that you actually would evaluate through ongoing verification, and if verification shows it's not working as you intended, then you would tweak it as you operate, modify the SOP. But when we talk about process controls, and in fact, I guess detection systems could be considered a process control, these are things that you certainly would validate because we're relying on the management of the parameters during process in order to control the risk. This is the part of ISO 22000, the section of ISO 22000 that talks about validation. And I just wanted to cite this to reiterate what some of our objectives are when we validate control measures. So ISO requires us to validate our control measure combinations prior to implementation of the control measures in our food safety system or inclusion of those into our food safety plan. We need to validate that the control measures are capable of achieving intended control of food safety hazard, as we've mentioned, that the control measures are effective and capable of, in combination, ensuring the control of a food safety hazard to obtain defined acceptable levels in end products. Control measures that cannot be confirmed need to be modified or reassessed. So if I can't demonstrate my control measure works, it just makes sense that I need to modify my food safety system in order to make sure it does. And we have a challenge when we look retrospectively at some controls in older processes that have been around forever and we've never had an issue, and we say, well, my validation is I've never had an issue. But that's a good input. That's a good demonstration that maybe something's working, but validation is intended to make sure we understand why that particular control measure or system is working, so we know that when we change that system, we can make the appropriate modifications to our programs, to our control measures. Modifications, of course, the reassessment, if we look at the regulatory language, reassessment or revalidation is important whenever we've had these modifications in our system that might change the way our control measures work. So I want to bring up this whole idea of risk-based metrics, and most of you have probably seen this before, but for me this is the best way for me to be able to sort out what I need to do for validation in a practical sense. So a food safety objective would be something that would be established by a regulatory authority. This would be the limit that they would allow, determine that it was acceptable to meet an appropriate level of protection at the point of the consumer. We don't have a lot of these to work from. Most often we have a performance objective, which is what a product or process should achieve at a certain point in the food chain. Often in the regulatory front, that's set at the retail level on the market with an understanding that what we would achieve in retail is somehow predictive of what we would achieve at the point of the consumer. As food processors, we often set our performance objectives at the finished product level as we're going into distribution with an understanding of how those distribution conditions impact the final level at the consumer, because of course we're most concerned about the level the consumer consumes, and so we need to be able to predict what our supply chain does or does not contribute to the product. So if I got a chocolate bar, supply chain is probably not going to impact the risk. If I have a fresh pasta salad, supply chains probably is going to impact the risk, and I need to take that into consideration. But this is a very good way to look at all of those control measures and how they work in combination. So we talk about the incoming raw materials. We might have a performance objective for those raw materials that we would state in our agreed upon specifications for our suppliers, because we know that's the incoming load we need to achieve in order for our system to work. And our system is a series of performance criteria, which may be product criteria such as water activity or pH or antimicrobials that are part of the product, or process criteria, which is some step in the process such as a thermal process or conditions of fermentation or other type of process in order to manage the risk in my process, maybe reduce the risk or itself prevent the increase of risk in the process. This is another equation that can help to understand how we evaluate all of the various control measures and the contribution of all the various control measures that we have in our system. And it's a way to assess and kind of calibrate the inputs that we need in order to determine what we need to validate, as we've been discussing. So this is an equation. Anytime I see an equation, I panic. I had a bad math teacher in school, and I was good at math. I think I was good at math up until like 3rd grade, and she ruined me. I blame it on her. So every time I see an equation, it makes me panic. But if you think about this as just a logic equation, so we've got H sub 0 that Don was mentioning. This is our incoming raw material contamination, and this minus well, let me go backwards. So we've set a performance objective, okay, so we know the level at the step of the food chain, which is a manufacturer. I'm usually looking at my finished product going out the door, right? And so I need to know that my initial levels minus the sum of all the steps I have in my process that can reduce the risk, plus all of those steps that increase the risk. That could be growth. If there's, say I've got steps in my process that growth can occur, maybe I'm holding a batter for a period of time in my operation, or maybe I've got some steps with semi-finished product before actually making the finished product, if growth can occur, or something we look at that's very important, recontamination risk from the environment. I need to take those into account, and I need to make sure that all of these factors working together are less than or equal to what I've established my process must achieve as a performance objective for food safety. So there's things that we can do to minimize the reliance on reductions. Often we focus on reductions. Often in food safety plans we say always when you do your HACCP study, know what your hazards are before you do your hazard analysis, because sometimes we pick a thermal process and we say that must be a reduction step, what is it reducing? And it may or may not be so important. It really depends on what's coming in on our product. But often we're looking at our thermal process or reduction steps, but there's ways that we can reduce our reliance on our thermal process or reduction steps. If I've got a process I've been managing for a while and marketing wants to make a fresher tasting product, or I want to save energy or water by reducing the processing conditions, I may be able to minimize my reliance on that reduction step or modify the parameters of that reduction step by managing certain things in my process. So I can manage maybe the initial levels. I can get more information on microbial levels and raw materials. I can also make sure that I'm picking suppliers that I know manage certain risks to the levels that are levels of concern. This seems like a no-brainer, but often we don't have a lot of good information about distribution of microbial contaminants on raw materials because we have a lot of presence-absence data, and we don't really share so much the data we obtain in industry, or even, frankly, often look at the data sets we have in-house to understand what that tells us about distributions. We just go to safe harbors for our reduction steps, and we don't focus on this information. But if I know my initial levels, I can make that determination of what kind of reductions I actually need versus just going with a safe harbor process, and I'll talk a little bit about safe harbors later. I can manage the increase due to recontamination through environmental surveys and history, and I can also, of course, manage the manage the processing environment to make sure that I don't have points in my environment that might increase the risk. This may be just environmental controls. It might be fill or a UV filtered or HEPA filtered filling zone to make sure that I'm not adding my hazard in the point of filling. So there's things that I can maybe do to manage recontamination. There's also things I can do to manage growth, and this could be managing our supply chain conditions, making sure my pasta salad only goes on a very highly dedicated supply chain where I know it's always under refrigeration, or I manage my shelf life to where I know my sandwich only has a 2-day shelf life, for example. I can also manage the conditions of reduction by more precise control over other factors to reach my performance objectives. So knowing I have a number of factors in my process that can impact my performance of my process, I may be able to impact the conditions of certain factors to manage other factors. So I may be able to manage the size of the container that I'm evaluating, or that I'm thermally processing, or I may be able to more closely manage the thermal process itself by having more information about the distribution of heat in a container or the way that my thermal process works. There may be ways I can tighten those conditions. If I have a better understanding of the thermal and activation kinetics of the organisms that are my target organisms in the system I'm looking at, then I may also be able to reduce the conditions of my process. I can incorporate more components into the process in order to manage risk. I can reduce pH. I can manage the product characteristics, for example. And I can, as I've said, limit variability in the process, and so reduce the need to rely on safety margins in my process. Okay, so where do we get our thermal process objectives? And a little this is a repeat of what was said earlier. The objectives for my thermal process can come from a variety of sources. So in a lot of cases, as Nancy mentioned, there's a regulatory standard that might outline the details of the thermal process. We have PMO that's pretty specific. We have, in the FSIS regulation, we have times and temperatures of cooking meat patties that's quite specific, and we can often just go and use that data without having to do much work ourselves, just demonstrate our process works. There's time temperature combinations for reduction of salmonella in certain meat and poultry products, also in FSIS regulation. There may be, instead of a specific time and temperature conditions for a specific product, there may be instead a performance objective for the process. So an example might be, an absence of 5 by 25 grams of Listeria monocytogenes at the manufacturer, and this would be, less than 0.008 CFU per gram, or less than or equal to 100 CFU per gram at point of consumption if I'm making a no-growth product in Europe, for example. It could be absence of E. sakazaki in 30 by 100 gram infant formula at any point in the food chain, or no more than 1 C. botulinum spore in 10 to the 12th container. So these are examples of where regulators, where food safety authorities have set performance objectives. It may be that a performance criteria exists where it's not the objective in the product, but it's the objective of the process that's outlined. So some examples are a process to achieve a 5 log reduction in juice, or a product to achieve a greater than 5 log reduction of salmonella in almonds, or 7 log reduction of salmonella in poultry products. These give us the flexibility to design the process in order to achieve the criteria. So unlike the low acid canned food regulation, they're not telling us you must run your process this way. They're saying there's many ways you can achieve this, but somehow your control measures or your combination of control measures need to achieve this reduction. There are other cases, in most cases, where there's not an industry or a regulatory authority stating what we need to do, but we as industry need to make things safe. And often we'll get together where there's something that's tricky and we'll develop an industry consensus document. We might create, like the ACMSF did, some default safe harbor values for Clostridium botulinum in chilled foods, 90 degrees Celsius for 10 minutes. We can go away from that, but if I'm a chilled food producer and I want to definitively control Clostridium botulinum, non-proteolytic Clostridium botulinum in those products, and I don't have a lot of time to do a lot of work, I know if I achieve this time and temperature in my process, this is a pretty good safe harbor that I can work from. If I want to reduce that, then I need to do more work, and I'll give some examples of that. And then we have this default for a Listeria cook that's been around for a while for Listeria in certain products. There are also cases where industry works together to create standards and best practices, such as the low acid, or such as the nut handbook, or some of the GMA work on low moisture foods, and some of the work IAFP is now doing on frozen foods and Listeria. And again, if there's no established criteria, industry is responsible for safety. And I just want to point out that there is no regulatory criterion specifying an absence of salmonella in chocolate. But we all know that we can't have salmonella in chocolate because of the risk. So I don't need a regulator to set that criteria for me, but to know that that's what I need to manage. And I don't manage it only because a regulator establishes a criterion for me. And in most cases, we don't have criteria that are very specific. So we have to figure out what those criteria are, as Don was talking about earlier. I threw in these slides because Katie had mentioned that we were focusing a lot on thermal processes, and I was a little guilty of that in looking at reductions in the processes. So just wanted to point out that there's other safe harbors for other conditions. If we look at the product criteria that prevent growth or work along with my other reduction steps to manage the risk. These two tables I'm showing you are from the food code, and these are also safe harbors. Experts got together, they said that if you operate within these conditions, and these are water activity values and pH values, then you can control certain hazards in your product. So this is the hazards of spores in foods that are heat treated to destroy vegetative cells and subsequently packaged. And the next table is for vegetative cells in spores in food that's not heat treated or that's heat treated but not packaged. So those are default values that you can work from, again, without having to do too much additional work in figuring out what those conditions are. And these, again, are in the food code. If you've not seen the food code, it's a great resource. You can just Google it. Okay, so just to review some of the guidelines, requirements for validation, and then I want to spend a couple of minutes on some specific examples of validation exercises. So in some cases, there's specific regulatory guidance on submission, on validation, but these are mainly related to These are mainly related to drug applications. So to date, there's not the kind of detailed regulatory guidance on validation from FDA that we have on drug and medical applications for food. But we can still use those principles. The European Hygienic Engineering and Design Group has some examples on continuous pasteurization, cleaning in place, and aseptic processing. This is an industry, a consensus of experts, and this is also Googleable. Is that a word, Googleable? It's also searchable on the internet. The Institute for Thermal Processing Specialists has some protocols for conducting validations that are good reference documents. This is the FDA guidance that I was referring to, and again, the principles here are very good principles to apply to any kind of process validation. There's also some other sources. The Almond Board in California, I think Nancy mentioned it earlier, has some directives on how to conduct validation. FSIS has a compliance guideline. I don't have the date. It came out in May 2013. And it's a guideline on how to validate HACCP systems, and it can be applied other places as well, not just FSIS products. GMA has a validation of cooking instructions for not ready-to-eat products. ILSI Europe has a couple of very good black and whites, and these are like white papers that they do. They're available online, and there's one on validation and verification. There's another one I don't think I referenced in my presentation, and it's how to develop alternative approaches to safe harbor thermal processes, and it also covers some of the whole chain approach I've been talking about here. Codex has guidelines of validation of food safety control measures as well, and we'll talk about that in a minute. There's some publications. Jenny Scott in 2005 has a good publication on validation, and National Advisory Committee has some guidance on how to validate alternatives to pasteurization. And of course, the experience of specialists working with a laboratory or other external group that works with the same kind of products that you've worked with that has some good experience with validation is also useful, because there's as much an art to it as a science. So very quickly, the Codex guideline has tasks prior to validation. I'll just whiz through this because Nancy covered it. We need to identify the hazards to be controlled. We need to know the safety outcomes to be required. They could be, as I've discussed, by regulatory authorities, industry consensus, or through our risk analysis of the product and process. We need to understand those points in the process, the performance objectives or the product and process criteria that we need to achieve in order to meet our performance objective. We need to then identify the points in my system where I need to validate those control measures. It could be a process criteria for a thermal process. It could be a product and process criteria related to hazard control. And we certainly need to look at whether or not this control measure has already been validated, because with all the work that we need to do here, it's silly to repeat work that's already been done, provided the matrix you're looking at and the process is the same or more conservative than what you're looking at, or a better case than what you're looking at. Some approaches to validation: we can look at reference, again looking at existing information, science and technical literature, previous validation studies and historical knowledge. And then, or we could, if all of that is missing, if all else fails, then we could generate valid experimental data to demonstrate the adequacy of the control measure if this doesn't exist. We could look at mathematical modeling to direct us to those control measures that, and minimize the validation work that we need to do. I would not rely on modeling as in isolation as my validation step, because modeling is as useful as the data that goes into the model. But models they get better and better with more data, but we need to verify, or we need to validate the output of the model to make sure that we're truly demonstrating the performance of my control measure. We can collect data during operation. We conduct product surveys as inputs. Okay. So this is from some guidance. I think this is from the FDA guidance I referenced earlier, and it just talks about the steps in validation, design specifications. This is where I'm determining what I need to do, and how my product and process needs to perform. So I would produce my design or develop my design criteria based upon these challenge studies, or based upon my literature reviews or industry standards, et cetera, as we've discussed. So this is my input. The outcome of that is my input. But then there's these other steps in the process to make sure my process is validated to be able to meet the conditions I've identified. And it's important we don't miss these in our validation work and our validation timelines, because we need to make sure that we're installing the equipment to function correctly. First of all, that we've selected the right equipment, that we've qualified them operationally to make sure we're achieving the conditions, and of course that ongoing they're performing as intended, and as we've talked about, change control is very important. So what I want to spend the last couple of minutes on is just to give you three examples that demonstrate surrogates, as we've talked about earlier, but also an example that demonstrates a really complicated validation and one that's pretty straightforward. So this is some surrogate data. I put this in from our last conversation because I thought that was a pretty relevant discussion here. This was some work we did with flour back in 2009. We were looking at heat-treated flour for our Toll House product, ready to our refrigerated cookie dough, and we did quite a bit of work with our partners looking at reductions of salmonella during thermal processing. One of the things we saw is that when you look in a dry system, many of you may know this, salmonella is actually more resistant than Listeria in a dry system. In a wet system, it's opposite. Listeria tends to be the organism we focus on in our validation. In dry system, it's Salmonella. And here we were looking at also Pediococcus, because we were trying to do some work to see if the Pediococcus strain would be able to work with us, if we could use that in our industrial validation. And I thought I had a table somewhere, but I've excluded the table. But basically, we found out that under the conditions we were looking at, that the Enterococcus strain the Pediococcus strain we were using was more resistant to heating than the Salmonella strain, so we figured that industrial applications we could use the surrogate. So when we set about validating, just to change to the two examples, my examples are related around some of the work we've done with our dry nuts and cocoa beans, because Nestlé is a company, we do a lot of chocolate. Sometimes that's all people think we do. We do a lot of products, but we're iconic for chocolate, and we've all known from some of the discussions in this meeting that dry heat, dry processes are very difficult and they're not as easy to validate as wet processes. So when we went about trying to validate our dry roasting process in one of our facilities, we were looking at some of the existing targets to try to determine what kind of reductions we should achieve. And we found the Almond Board of California work, talking about a 4-log reduction. We saw that FDA had indicated a 5-log reduction for peanuts and pistachios. And we had internal data that showed that Salmonella was frequently isolated from raw cocoa beans, but at a level less than 1 CFU per gram. So we used this in order to determine our targets, and our target was a 4 to 5 log reduction in our process. The GMA Nut Handbook gives us some guidance on how to conduct such a validation. They talk about the surrogates being necessary for factory trials, and they also indicate that we should be comparing the conditions that we have with some published data, so we're not repeating work that's already been done. But unfortunately, there wasn't a lot of work already done, so we were on our own. Oh, this was the table I got out of place. This is the table showing the surrogate. This is 13% moisture, 7% moisture in flour, and you could see the surrogate had a much longer decimal reduction time under these conditions, so we were able to see that the surrogate was appropriate for our application. Okay, so this is cocoa processing, and in cocoa processing there's a couple of steps where heat's involved. So we know there's Salmonella coming in on the cocoa beans. We want to know where in our process we have that ability to deal with Salmonella. And here I'm talking not just on environmental control, because we know that environmental controls, water control, zoning, those are all critical. But in the process itself, here's the steps. It's roasting, okay, and conching is another step where heat's applied. So we looked at conching, and conching is where the cocoa is ground up into a paste. That's a very oily paste, and there's a lot of heat. It's a very intense process, and there's quite a bit of heat. It gets to about 50 to 70 degrees Celsius, but it's a dry heat, and we saw very quickly that we weren't getting much of a reduction in the conching process, even though it's dry heat for a long time, even though it's a relatively high temperature for a long time. So really the best we could rely on is roasting. Roasting became our CCP in our operation, and we had in this facility 2 roasters. And it was a dry roaster. It was a spiral roaster which pushed air through the product stream as the product was passing through the roaster. And it was an old roaster, so but still made very good product. And here is some showing you what we were up against. This is some of the GMA summary from the GMA low moisture foods documents. And you could see that in chocolate we were seeing at 70 degrees C, 440-minute decimal reduction time. Even at 90 degrees, it's still 36 minutes. So we're seeing a pretty hefty request for the performance of our roasting equipment. But we do roast at pretty high temperatures. So what we did is we selected strains that were typical. We selected strains that had high heat resistance, and a couple of strains that were related to chocolate or actually chocolate and raw nuts that were related products. And we did a couple of different trials. So we started off wanting to do a nice trial in closed vials. So we looked at cocoa, we looked at hazelnuts. We took a lawn of Salmonella cultures. We pooled it in an oil slurry. We inoculated a shell powder from the nuts and almond shells, and also from cocoa shells, and we heated them in glass vials. This is the easiest way to do it, and here's what we found. We got if you looked at the average of trials, we got pretty much we could tell what the decimal reduction times are at these various temperatures. The problem is we had a lot of error and we had a lot of variability, and if anybody's done vial trials, you know that there's a lot of reasons why you can have variability in these trials, and we saw that the data just wasn't data that we could use, so we were back to square one. So what we did is we knew by the way that we couldn't actually inoculate the roaster. There was really no way to do that. So we created a bench scale coffee roaster, and this is what it looked like. We tried the best to replicate the conditions of the roaster, and then we were able to measure the bean temperatures by sensors, and I'll show you a picture of that. But first of all, we irradiated cocoa beans and the nuts that we were evaluating. We did different trials with cocoa beans and nuts. We grew salmonella cultures on nutrient agar. We harvested it in BPW. We added it to the material. We massaged it in a stomacher bag, and we dried it at room temperature for 6 hours. And then we came up with this crazy device to actually embed the thermocouple into the nuts. This is what you need graduate students to do, I guess, to figure out how to do this. You need that ingenuity and focus. So, but this allowed us to figure out the conditions of our small-scale roaster, and because of that, we were able to mimic the conditions in the roaster. So we did trials on these nuts to make sure that the come-up times and cool-down times were similar, and that we had the right density and able to mimic the conditions of the roaster. And from that, we were able to successfully get some decimal reduction times by analyzing product in this tabletop roaster and pulling product at various times. And so in the end, we were able to evaluate that for cocoa, we get at 120 degrees, we got 5 minutes. In almonds, we got 9-minute reduction. Okay? And then we were able to convert this to a market policy for all of our factories because we'd done that work. Very quickly, though, the problem with that is that took a lot of work, and then marketing and product development says, you know, it'd be great if we could change the roaster conditions because we'd really like to have the nuts with different conditions. And we're like, oh my goodness, this took us a long time. What do we do? So what we said was continuing to validate and change the roaster conditions in dry roasting was too complicated. So we looked at something called a debacterization system, and what this is, it has a little chamber, the cocoa beans go in the chamber, and then there's steam that goes into the chamber, and it's superheated steam. It's a continuous batch process, if that makes sense. It's small batches, it's very quick, and so you have in the steam, here's a steam reactor, it gets steamed in the hopper, and then eventually this is my old dry roaster. Okay. The way that it works is it's superheated steam, so the shell gets heated to a high time and temperature, and it's a moist heat, but it's so quick that it doesn't really get through the cell, and you have to cook the beans in the shell so it doesn't get into the inside of the kernel of the cocoa bean. And so we were placing the debacterization unit prior to our roaster, and then that we wanted to become our CCP. So we used conditions that were stated as typical for our products by the manufacturer. We were targeting a 5-log reduction of salmonella, but we also knew that some of the literature looked at spores. So we said, okay, well, if we get a reduction of spores, that should be enough for salmonella. We did 5 representative blends, 10 trials, and we looked at the samples before and after debacterization for spores. We weren't even looking for salmonella. We were using index organisms here. And then we did 5 by 300 grams per trial. And just to show you, this is the data, sorry for the table, but if you look at, this is the actual numbers, this is the log values. But we were getting for spores, if you look at thermophilic and mesophilic spores, we were getting a 5 to 8 log reduction in thermophilic and mesophilic spores. I don't need to put salmonella in there to see that that's working. Okay, so our total counts were going hundreds of thousands per gram to less than 10 per gram after we used this unit, and then that allowed us to have some flexibility in how our roasters worked. And this is just the manufacturer data showing us what we learned ourselves. So, a couple quick examples. I guess to conclude, just to say, don't reinvent what's already out there. Make sure that you're looking at existing literature, and make sure that what exists, what you're using, represents the conditions that you have in your product and process. You'd like to avoid doing these kind of studies if you can, but there are opportunities if you have a difficult process to work together with others, with peer companies that have the same problem, maybe to generate data through like GMA or IAFP or some other organizations. So thank you.